Got it. Mhm. attested to his dedication and leadership in the academic community. He's fellow in the national system of researchers and has over 53 scientific publications with over 4,970 citations, according to Google Scholar. It is an honor to have you here today and hear about his groundbreaking research. Please welcome Dr. Cruz.
Thank you. I hope you can hear me in the back. Okay. If not, just let me know. I tend to lower my voice, and I also want to thank the organizers for the invitation. It's been a couple of yesterday and today, very nice welcome by the students. Also, I apologize for my Spanish. I'm a little bit rusty, so excuse me for that. And I will try today to show you what we have been doing in the past 8 years in our laboratory. First, from a very global perspective to a very tiny and molecular detail, how we can use animal models to answer scientific questions. Okay? So the next one, please. So when we talk about the omics, we talk about different technologies that are next generation technologies, which are genomics, proteomics, metabolomics, which is profiling at different levels of organization the information of a certain animal or certain process. So which is the animal we studied in the laboratory is the axolotl. And Ambystoma mexicanum is this guy. Everybody knows the white version, you know, the albino version, but this is the real original one. And of course, it's been a very famous salamander all over the world, not only in science, but also in graphics, in music. You will see it's very famous in Japan, in China. It's very charismatic organism. But what are the axolotls or the apolotls? So this Ambystoma mexicanum belongs to a larger genus, which is the Ambystoma genus. And the Ambystoma genus is composed of 32 species, which live from South Canada till Veracruz, Mexico. From these 32 species, there are 16 species which are endemic of Mexico. And only one of them is Ambystoma mexicanum. So why Ambystoma mexicanum is so famous? Because Alexander von Humboldt, several hundreds of years ago, when he came to America, to the continent, he started to collect samples from all over the continent and then brought them to Europe. And then they realized that the axolotl has this remarkable capacity to regenerate. So that's why, since those days till now, it has become the model, the king of the vertebrate models to study regeneration. There are other organisms that also undergo regeneration very efficiently, but they are not vertebrates. Okay, what else? The next one, please. So these are the 16 species that inhabit in Mexico. The axolotl is only one of them. I think it's that one. And if you can see, some of them, they are alone, and some of them, they have a salamander, a terrestrial salamander next to them. If you can see, for example, helix, no? That's because some of these species undergo metamorphosis, while others cannot. So those that cannot undergo metamorphosis are called neotenic. And the Ambystoma mexicanum is a neotenic. So it never becomes a terrestrial salamander, even when it reaches its adulthood. While the others, when they reach adulthood, they will become terrestrial salamanders. And some of them are depending on the weather conditions. If there's more heat or less water, then metamorphosis will be triggered. So that's another very interesting characteristic of these genus. Not only regeneration, but also the capacity of metamorphosis, a variety capacity of metamorphosis. The next one, please. So this is a gift of one of our students. So we can follow from Ambystoma melasi, a cousin of Ambystoma mexicanum, how in few days it can become a terrestrial salamander if we apply a thyroid hormone to the medium. So it completely loses the gills, the tail reduces, it starts to breathe through the nose, and because before it was breathing through the gills and the skin, and the heart and the lungs develop to live in this not aquatic condition. So that's fantastic, because we compare this to in humans, we live before birth in an aquatic condition, and we have to develop our lungs and our heart before we cry, no, we shout, and then there we need to breathe the air, and all the organs not need to be perfectly developed. So that's another point which is very interesting to study here in metamorphosis in this organism, how the lungs or the heart develops in order to function and changing from an aquatic to terrestrial condition. Okay, so the next one, please. But the main question is regeneration. So all over the world, and when I say all over the world, is literal. In China, there are axolotl, in Japan, in all Europe, United States, there are 50 research groups all over the world studying just regeneration using Andistoma mexicanum, and only from 2 or 3 events after Alexander von Humboldt, and then 2 or 3 more events, they take out the animals from Mexico, and then they breed it, and then they get these mutants which are albino or the golden ones. There are these 3 different mutants that they lack the color, the black color. And all these laboratories, including ours, we are studying why these guys, they have the huge capacity to regenerate not only the limbs, but also parts of the heart, the gills, the eyes, and perhaps many other organs that we haven't tried, like the pancreas or the liver. But the main question is why they do it and we cannot regenerate, or we can regenerate to a certain level, but we never reach the level they have. And we need to answer those questions at different levels of organization of the molecular information, from the epigenetic, genomic, the transcriptional, post-transcriptional, post-productional, and now the metabolic. So if we want to understand a phenomenon, a biological phenomenon, we need to apply now all these questions to answer it, especially if it's developmental biology. The next one. So why they do it, why we don't do it so efficiently? And if we could learn how they do it, maybe we could apply this to biomedicine, to human health. So in our lab, we are very interested in understanding how it works, neotenic metamorphosis regeneration mainly, and also we want to support a little bit with this information the conservation of the earth. The next one, please. So I will move to start. As you know, 10 years ago, we didn't have a genome in the archaeology. So it is the biggest genome ever sequenced from an animal, 32 gigabases per 10 times bigger than the human genome. So it needed several million euros to be decoded, and the work of at least 15 laboratories all over the world. We were not the leaders, of course, because we didn't have the money, but we were invited to collaborate. And in 2018, the full genome was sequenced and published. And maybe you know that there is now a National Day of the Axolotl, and that's 18th of February or something like that. And that's because that's the date when the genome was published in 2018. So 2018, just 5 years ago, we didn't have a genome. Now we have a genome. But when you have the genome, we don't have all the information. You need to see how this genome is expressed, is transcribed. Now you all know big molecular biology, and the genome becomes transcripts, and the transcripts, some of them will become proteins, some of them. Then the proteins will do a lot of work, and maybe they will process like enzymatic reactions, and they will produce metabolites. So what we are doing is trying with many other laboratories in the world is not only to get the genome, but also the transcriptomics, and also to get the metabolomics. Why we are doing that? Because we want to understand the small pieces that conduct the regeneration capacity of the axolotl. And then maybe we can learn how they do it, and maybe the doctors someday will do it. Okay, the next one, please. And more recently, we participated in this new technology, which is single cell transcriptomics. So you isolate only one cell, you disaggregate all the cells of the organism, in this case the axolotl, and then you mark them with small barcodes, and then you will sequence in each cell which transcripts are being expressed, which mRNAs are being expressed in that moment in that cell, and then you can do those maps that you see there, and then every color in those maps will belong to a certain type of cell. Let's say the liver or cells from the blood with a specific marker genes. Okay, so that's what we have been doing in the past years, try to collaborate to get a lot of information, massive information, the omics. Without this information, we cannot do too much. We will progress very slowly as in the past. The next one, please. So this is blood. Okay, so you have the amputation of the limb, and then there's a closure of the wound healing, it's called. And then there's information, metabolites, that tells the cells that are behind the cut that they need to reactivate in proliferation, start to divide again, to form this structure called the blastema. And then this blastema will grow, and then these cells will have memory, and then every cell now will become a muscle or a nervous system or chondrocyte, and then will form the whole new limb. If we can go to the next one, please. This is the real life. This is Mexicano Velasquez, and we try with both, and then you have the intact limb, and then we make the cut in a specific region, and then we follow what happens in the real life. These are adult animals, more or less 18 to 20 centimeters. In 75 days, you will have a new functional limb, which is fantastic. And still, it's amazing, many labs in the world. How this happened, that's what we want to understand. Please, the next one. So this is more or less more Mickey Mouse, I say, what's going on after the amputation. So after the amputation, there's the wound healing, the wound epidermis needs to be formed again. And at the same time, there are molecules like calcium, ATP, reactive oxygen species, and other molecules that will send signals to these cells, which are differentiated, part of muscle or other tissues. And then these cells, they will start to lose and to become more proliferative. So they will reach a capacity to divide more rapidly, but they will never lose their identity. And they will form this structure that is called the blastema. Okay, and then after the blastema has grown, then this cell remembers that it's from this tissue and starts to reorganize in the new tissue. Okay, the next one, please. But how this happens, how a cell that is fully differentiated, not dividing, now gets the capacity to divide. No? And maybe you know from this Nobel Prize in 2012, I think, the Yamanaka experiments, that they used these mature cells, and then they made a cocktail of 1 or 3, 4, 5 transcription factors, oxox, nano, MIC, and also LID28, and then put it in this cocktail of cells in the culture, and then all these cells will start to become pluripotent stem cells. And now there is engineer of tissue, so you can create new muscle tissue. There's even an experiment that they created synthetic meat for hamburgers. That's for fun, but it's true. No? But now we can create engineer of tissues you can create making all these cocktails, and you can drive the cells to become what you want, to divide and to create new tissue. So based on this, one of these major factors of proliferation is Lin28, which is going to be the next part of the talk. It's going to be really, really molecular. So please don't lose the tail of the lizard, because then, well, maybe in the questions I can answer, but try to be focused on Lin28, please. The next one. So what is Lin28? Lin28 is a protein that is conserved in humans and many other animals, and it's a protein that binds to RNA. So is this one here? So it has two domains that binds RNA, and there are two Lin28s, A and B. One is localized in the nucleus, the B, and one in the cytoplasm. So what we found, Hugo found, one PhD student in our lab, that there are also two Lin28s in the axon, Lin28A and B. And these Lin28A and B, they bind to a family of precursors of microRNAs. So microRNAs regulate the expression of transcripts and the expression of proteins by binding to its targets. So what we have here, it's a loop, a negative loop of regulation, in which Lin28, by binding to let's 7 microRNAs, degrades the microRNAs. But if there's some let's 7 microRNA, it will bind the transcript. Where is this? Somewhere here. It will bind the transcript of Lin28, and it will degrade Lin28. So it's again, if you break the balance, then you will go from a proliferative state to a differentiated state. The next one, please. Why they are important? Again, because if you have high levels of Lin28 and low levels of let's say 7, you will have stemness, proliferation, which is what happens in the blastema. But when you have higher levels of let's 7 and low levels of Lin28, you will have differentiation, which is what happens after the blastema has grown. The next one, please. So some experiments before, what they did is to overexpress in the mouse Lin28 in a transgenic line. And normally in mouse, if you cut the fingers, they will not regenerate. But now if they overexpress Lin28, then it can regenerate. You can see the next one. So you will see how they regenerate. And this is accompanied, the previous one, please. And this is accompanied with a lower expression of let's 7. So you have higher levels of Lin28, more proliferation, regeneration, but that means that you have to turn off the let's 7 microRNA. The next one, please. So this is Hugo Varela's project, a very talented PhD student. So he took all the information that I just explained, and then he started to explore this in the axolotl. The next one, please. So again, he found that there are two Lin28A and B in the axolotl, one which colocalizes in the nucleus and the other in the cytoplasm. It's very, very conserved in evolution. The next one, please. So that means, what means that it's very conserved in evolution? That probably it will have more or less the same function as in humans. So which it will be either to trigger proliferation, or if you go to the let's 7 balance, a higher levels of let's 7, you will have more differentiation. So what he found is that in the genome of the axolotl, there are all these loci that are encoding the precursor of the let's 7. These are all these let's 7s, the precursors in RNA, and this is all the conservation in base pairs. So you can see that it's really, really conserved. So there are all these loci coding for 11 pre-microRNAs and 8 different mature let's 7 microRNAs. The next one, please. So we have both guys. Lin28, let's say, okay? But then what happened? So he then checked what's going on with the expression levels of Lin28A and B during regeneration. And then he amputated and then extract the RNA and then follow the levels of expression. And you can see that Lin28 expressed highly in the blastema stage, really, really high, while the Lin28A
didn't change too much. But also it was very interesting to see that while Lin28 was increasing, Let7 during regeneration, Let7 was going down. So far, so good. The next one, please. And when we make sections and then we follow the regeneration process, and then we did some immune staining of Lin28 to see what's going on with the protein. What I showed you before is that the transcript is highly expressed during blastema formation, but we wanted to know what's going on now with the protein. So you can see that at certain point, you will see in the red signal is Lin28B, localized outside of the nucleus is in blue. But when you follow regeneration, you will start to see that Lin28 starts to introgress into the nucleus. So that was a major finding that was not reported there. More or less at 14 days when the blastema is already formed. The next one, please. And then if you keep following, this expression is even higher in the nucleus. You see less expression in the cytoplasm, but more expression in the nucleus. And then when these cells start to redifferentiate to form the new fingers or the new tissue, you have a confetti localization. Some of them, which are still proliferating, will be in the nucleus, while others will be more in the cytoplasm. The next one, please. And it was reported that in order to proliferate, cells need to have Lin28 inside the nucleus to inhibit, let's say. That was, and there's another protein which is called merlin, which is maintaining Lin28 in the cytoplasm in cells that do not divide. While in cells that divide, then merlin lets go Lin28, and then it comes to the nucleus and inhibits Let7 action. The next one, please. But then, what is this one? Lin28A. Okay, Lin28A is not that important because what we found is the green signal is that it never changes localization or expression level. So Lin28B is the actor of this plane. The next one, please. So we wanted to know what happened if we now block the action of Lin28B, and then Hugo used this inhibitor, the 1632, very creative name, 1632, and he used this technique where he put the inhibitor in a PCR tube, and then after amputation, he put this organism to rest, to sleep, and then put the amputated arm in the tube, and then see what's going on with the regeneration. The next one, please. And then what he found is that after application of this inhibitor, he found that there was a partial inhibition of the regeneration that could only be observed if we see that regenerated area plot there, that there's a mild effect. But remember, we have to put this inhibitor not all the time. We have to put it 1 day, every 2 days, and then in some point we'll see some effect. But in order to really demonstrate the function of Lin28B, we needed to create a full loss of function of Lin28B, which if we try to do that by transgenesis or something, maybe it will be little because Lin28 is essential for that. So we couldn't do all it but this approach. Okay, so far more or less good. The next one, please. But when he applied just by this mild application of the inhibitor and mild effect on the regeneration capacity, he already could observe a change in Let7 levels. So Let7 levels increase when we inhibited Lin28B. The next one, please. And okay, so again, the mature Let7 correlates with increasing Lin28B. But if you remember that Let7 also binds to the messenger RNA of Lin28B. So if we now are inhibiting Lin28B, we will have even more Let7, and then Let7 will still now the remains of LID28. And that's what he found. So he found not only a downregulation, an upregulation of LID7, an effect on regeneration capacity, but also the downregulation at the transcript levels of the LID28. The next one, please. And when he applied these to the animals and then made the sections and followed them with the antibody, using this antibody, again is the red signal, this is the wild type, and you see again the blastema, and then you have all these cells with this patchy red color in the nucleus and in the cytoplasm. But when he applied the inhibitor, you have an effect in blastema formation, as here, and then also a low expression of the protein. Okay, the next one. And then we were not happy with the previous experiments, and then we applied a more constant inhibitor, LID28B, and now you can see, especially in C, in the panel C, how it really affected the process of regeneration.
So what we have found now is a circuit composed of LID28, B, and LED7 that was not studied in the context of regeneration. And we found that in the axolotl, these circuits control the capacity of regeneration. What is fantastic is that we humans have LED7 and LID28. What would happen if we are able to modulate these molecules after amputation of a lead in the human? Maybe we could trigger regeneration. Okay, the next one, please.
It has been reported that LID28 and LED7 modulates the production of metabolites and the function of the mitochondria. I am not a biochemist, but what gives people power is mitochondria. And the next one, please. So normally, in differentiated tissue, the cell obtains the energy through oxidative phosphorylation. In most of the ATP comes from this pathway, and in lower extent from the anaerobic glycolysis process. But in cancer, proliferative tissue, the cells produce even less energy, but they prefer to use, I don't know why, this pathway to obtain energy, which is aerobic glycolysis, which is also called the Warburg effect. It's a Nobel Prize also of Dr. Warburg. And we wanted to know if it was happening in the axolotl. The next one, please. Next one. So what we did, it was a metabolomics now. We already show about the genomics, transcriptomics, microRNAs, all RNA level, and now we are going to the metabolite level. So we profiled in different stages of regeneration, extracted the metabolites, and then run them in mass spectrometry in collaboration with another group. And what we found, can we click the next one, please? So if we make this zoom, we started to see that compounds which are related to this TCA cycle were changing. If you can see, they were decreasing in these stages and then they increase in quantity. This is only metabolites, but this is called oral tension, no? And also many other metabolites as here, which we haven't explored in depth, like some phospholipids and glycerolipids. The next one, please. Okay, so again, so pyruvic acid, oxalacetic acid, and other organic compounds, acids from the Krebs cycle are changing during regeneration. The next one, please. And then this was a non-directed metabolomics approach, and this is a directed metabolomics approach where we measure what's going on with certain molecules, which are these three carboxylic acids. No? And what we found was quite interesting, is that During proliferation, in the formation of the blastema, all these acids go down, while in the differentiated cells, they are up. So we are reprogramming the cell not only at the transcript level, but well, we are not. The axolotls, the cells of the axolotls are being reprogrammed during regeneration from one program to another, but now it goes deep down to metabolites. And now coming back to the inhibitor of Lin28, we applied the inhibitor and we found that this behavior was not followed. So basically Lin28 and Lin7 is controlling upstream. We don't know how, maybe by controlling some enzymes here, the levels of 3-carboxylic acids, and this is quite important for proliferation and differentiation reprogramming event. The next one, please. That was our work, and then this work came out 2 years ago, and what they found, please believe me, the red signal is mitochondrial signal, and the green signal, what red signal? Green signal, and then what they found is that during regeneration, there are more mitochondria formed in the blastema. So, the next one, please. So that correlates with what we found. Every arrow here, it's an extra mitochondria found in the blastema formation. So this correlates with our work that not only we have a change from oxidative phosphorylation of the cells to obtain the energy, but to aerobic glycolysis, but there are a lot more mitochondria being formed in each cell of the blastema. The next one, please. And this is more or less, because I don't want to bore you too much, a resume of what we found and other groups found, is that when we have a higher capacity of proliferation, we have more Lin28B, which downregulates Lin7. And when we have a differentiated tissue, Lin7 levels are higher, which downregulates Lin28. And this has a very nice correlation with glycolysis. When there is more proliferation, we have aerobic glycolysis and more mitochondria producing all these effects, but a lower oxidative phosphorylation and less products of the TCA side. More or less, we try to simplify all the big puzzle, but that's the best we have done so far. The next one, please. And finally, before the questions, maybe I want to, in fact, but I want to thank all this work was, you can tell it in 40 minutes, 30 minutes, but then it's work from several people from 8 years. So I want to thank all the students, Janet, working on the metamorphosis. I didn't show the results of Janet just for the introduction. Most of the work in Lin28, Lin7 was done by Hugo Varela, and Cesar Garcia was also helping, and technically Alice Pinal, and computationally Juan Caballero Perez. And also want to thank all these collaborators because many things we did in our lab, but many others did in other labs, specialists not only in Mexico, but other places, and also the people who supported with the conservation and providing the animals from Juan Xochimilco, Mania, Corena, and also the money we obtained from all these institutions to make our research work. And the next one, please. I think this is a commercial of 2 postgraduate programs that we have in Irapuato, in Simvestab. Simvestab is a network of 12, I think, research centers that are the major producers of science per capita in Mexico, even higher than UNAM. I don't say this at UNAM because I will be killed, but we have these 2 postgraduate programs. 2 of them are from International Excellence. This one has 30 years, and I think it's the top in Latin America. And this one is very new, more integrative, more multidisciplinary. And of course, if you are interested and soon to graduate, the call will be out in March. Are we March? No, tomorrow. Maybe from tomorrow onwards, you will see the call in those networks. And I think that's all for me. The next one, I am open for. Thanks a lot.
Question, guys?
When you cut out the link, you try to do it at different levels of the lid, like if there is a point, how high you can cut it up when you break it.
So these are pioneering work from people in the field. They have cut in different regions. And they have shown that you have to cut either here or here. Because if you cut here, because it's a nerve dependent process. And then if you cut here, then tissue will retract. And this, the region of the lesion, the nerves will not reach the region of the lesion. And you need the nerves. So you need to cut here, but not near the yeah. Otherwise, it's not.
Question. Thank you, Dr. Cruz. It was really interesting your talk. I enjoyed it a lot. You mentioned already that there is some similarity with proliferating cells and cancer cells. Does anything in your research have given you any information how proliferating cells in Axolotls avoid becoming cancerous? Or I know it's a complicated question, but have you seen any?
No, it's quite interesting and it's complicated and I'm still figuring out how to use the axolotl as a model system to understand things from cancer, because it has been shown that if you apply carcinogenic compounds to the axolotl, it will not become created. So we want to know why. So far, nobody knows exactly what's going on there, but many things reported as stem cell or stemness factors are also known as cancer factors. So if you mutate whatever of the genes in one side or the other, in the mutation, they are also reported to have a role in cancer. So why the axolotl cannot undergo refractant, I would say, I don't know the word they use, but there's a refraction to produce cancer, we don't know, or we don't know. So far, many people want to know that, but very few laboratories, I don't know why they don't want to mess with that. But yeah, there are many genes related to cancer are also, they are deregulated in regeneration. I didn't answer your question because it would be great to understand why.
Could you speak louder, please?
Is that also reversible? Like when you stop the regeneration process and then can you like make it regenerative again?
So if I stop with inhibitor regenerate and then remove, it can regenerate again. Yeah, it can regenerate because as you see, we didn't put it all the time. So it still regenerates, but it has effects. If you fully downregulate by a knockout or full knockout of the LIN28, I think would be very difficult to have. Then I think we haven't done that experiment, but I imagine that it will be reversible. It will lose the capacity. Because it needs this LIN28 to proliferate, the cells to proliferate.
You mentioned that actually also regenerates organs.
Yeah.
Have you tried studying the pathways that organs regenerate, or did you see?
I haven't there to do that because you need to have knowledge on surgery. Yeah, it's difficult to do that. Some labs they do it. They for example, cutting certain region of the heart, of the liver, And they haven't tried in 28, and it doesn't work there, because we just published that paper in 2020, but maybe they will do. And I guess that it will be more, not only in the limb, but also in the heart and in the liver. We don't do it because it's quite difficult for us. We just focus on the limb or the tail. That's what we do. If we don't have the knowledge or the practice, and you need to be certified to manipulate the animals in that sense. Maybe some point when we have more money, and people train for that, we will. More questions?
How far are you from trying something in humans? How far are we from making some?
Less than yesterday. And we need to give crop. It's a question that always pops. And what I have to say is that we are closer. But to be honest, I will lie if I tell you 20 years. But maybe 20 to 50 will be not that far. We will still see this. Maybe just to apply, how could we apply something that I think that in some point we modulate these molecules, trigger the regeneration, and then you take out those guys because then you can trigger cancer. So that's the main challenge. Now we have some molecules we need to try, but I guess to try all these molecules now in mammals, which are models, will be even in 2, 3, 5 years. That can be done. But in humans, I guess it's gonna take longer, maybe in 20 to 50. I will get.
Yes, I'm not biologist, so I find that your presentation really interesting, and something that is just going in my mind is that you have mentioned that we humans, we have this LIM28 and this LIM7, but why in humans we don't have this capacity of regeneration, or maybe we have it, I don't know, but not as they have it. And the second question is, because you have mentioned that the animal is capable of regenerate functional limb, but to what extent? Is it 100% functional, or there is a level of functionality that is lost during this process?
Very nice question. So first one, we all have the capacity to regenerate. Perhaps if we could go down a couple of slides. I was not prepared for this question. Next one, this one. This is evolution of regeneration. So if you go to planarians, to invertebrates, they have the blue is the capacity of regenerate, and they are able to regenerate the whole body, sections of the body, and they are really the champions in regeneration and the invertebrates. And then if you go through evolution, here is the axolotl, and then the birds. So the birds, they have almost lost all capacity of regeneration, while some salamanders they keep, but they lose the capacity of regenerate the whole body. And humans, we have the capacity to regenerate hair, skin, the liver. So I guess for those processes we need the LIM28, the 7 also to be conserved. But why we don't have why the axolotl has lost the capacity to regenerate the whole body, and we lost the capacity to regenerate limbs. And that can only be explained by evolution and linearity. And what I think is that if we were able to regenerate, and we are exposed to a lot of different environments, the sun, contamination, etc., the levels of cancer in the human population would be higher. And that some people say, we lost the capacity, we are less advantaged. Maybe we gain that capacity of not regenerating. We pay the price of losing capacity of regenerating to protect ourselves. That's one hypothesis. And the other one is how
100%, they can do it normally. If you cut it one after the other, of course, there will be a problem with the teeth of the animal. But only if we just cut it once and then we in a safe place and it's healthy, the link will be 100% function. It will swim again.
Thank you, yes.
So, maybe the public video on regeneration episode is a bit far-fetched today, but what would you say about limb reattachment? There's a lot of patients that go have problems with their damage or maybe internal tissue like organs. Like how plausible do you think that it would be? You could that people could develop treatments for this kind of problems within technology.
That's a good question. Also, there are some attempts working, especially with heart regeneration. So if you cut the heart, just you see the heart is there's a tip in the heart, and then you got this tip. This is the region that you can regenerate. Of course, when you have all this connection, the wires and cords, then if you got there's no regeneration because you need to have blood pumping and many things. Then they cut it down, and it's able to regenerate. And they have been done this in several fish and the Axolotl, and they've been trying to make some gels. Now this nanotechnology generating some gels and putting in these gels some molecules, and then make the surgery and put in that region of the damage these molecules and see it increases the capacity of regeneration. So I guess at some point, if we are able to put a new heart or an artificial heart in a human, perhaps we can now use these gels. Of course, do the surgery and do the gels and then manipulate a little bit the molecule. So it could be as feasible as in the really good talk.
Yes. Joe's first. Is the capacity of regeneration in Canada, for example, the Sigma, the one in Axolotl?
Sorry.
The capacity of regeneration in the kind of is it the same as in Axolotls?
In the what?
In the kilograms,
The kilograms. No, it's even higher. It's even higher in the kilograms. So in the invertebrate, like the swans, it's even higher. So we have, the sea stars, I think it's in the kilogram. And that's also a very nice model for regeneration. And there was another thing.
Do you know if these pluripotent like mechanism can be reproducing in vitro or only do you need the actual animals and breed?
No, it can be reproduced. I mean, the Nobel Prize was only using human cells. Yeah, and also that's the way we have these animal clones. That you took the nucleus from a ovule, and then put it in the region of the nucleus of a major cell, and then you can create a new embryo. So this has been done in Dolly, to clone dogs, to clone sheep, even clone human embryos. So that can be done also. If you take it in a cell culture, you can also put these cells and then do all these proliferations. Right. Thanks a lot for the cure. There is no questions online. Thanks a lot.
Appreciate your participation in this.
So that's the Utrecht. Oh,
yeah, see presenting next.
oh, cool.
Recommendation list. Yes, and gifts for the AIDS Institute.
Thank you very much, all of you, and sorry, thanks a lot for the invitation. Very nice camp you have.
So, guys, if you haven't registered yet for this one, the notebook was going around. And if you're staying for the next one, we'll start another, page just for the people attending the next program.